movement enters the cecum directly. So this is the cecum, and then the cecum connects with the colon. So in that case, here there may be a valve. So this is ileocecal junction. But in some species, the cecum and the colon, ileum enters here. So what is this? Is what cecum, colon, ileocecocolic junction, right? So there's a junction here. So this is so at the junction of the cecum and the ileum, that's where cecum and colon, that's where the ileum enters. So this one is directly on the cecum. So I want you to give me the species. You know? Equine. And
Lagomorphs.
Humans too. So rabbit. So check. So give me the which animal species have this and which animal species have this tonight. Submit it to me. That's your anatomy. The simplest thing, just call your anatomy professor and then he will tell you and then he forward it to me. All right? Or you can send him an email. Quickly, he will give you the answer and you forward it to me. All right. So, the recto-anal junction, of course, is just the junction between the rectum and the anus. So, here we will still get to the physiology. So, functions of the large intestine. Of course, major absorption of electrolytes takes place in the cecum, in the ileum. However, some electrolytes are also absorbed in the large intestine, as well as water. It is also a fermentation vat for most of those materials that are indigestible by the host enzyme, that host intestinal enzymes. So here you find microbes in small amounts, but in hindgut fermenters like the horse and rabbits, there is massive microbial population of the large intestine, whereby fermentation is the major digestive mechanism in these animals. So they have intestinal enzymatic digestion, yes, to a less degree, but the major breakdown happens by fermentation in the large intestine. In fact, it's been demonstrated that the fermentative capacity of the large intestine in hindgut digesters is almost the same thing as in ruminants. In other words, what is happening in the rumen is almost the same with what's happening in the large intestine of horses and rabbits, to the extent that the microbial biomass and the volatile fatty acid production in the rumen and in the large intestine of these hindgut fermenters are almost the same thing. So for every 1 meal of fluid harvested in the rumen, compared to every 1 meal of fluid harvested in the large intestine of hindgut fermenters, if you check the microbial population as well as the quantity of volatile fatty acid, it's going to be the same. I'm mentioning volatile fatty acid because the major product of fermentation is what? It's taking place in the lumen. And it is coming from, it is carried out by what? Pancreatic enzymes. So we still see the other type of digestion that will now break it down finally. So the pancreas is a very large compound gland which lies parallel to the stomach and beneath the stomach. Parallel to and beneath the stomach. What does it do? It secretes digestive enzyme and large volumes of sodium bicarbonates. I just mentioned that. Sodium bicarbonate coming from the duct cells, and then digestive enzymes coming from the acinar cells. So now again, These cells also, how is bicarbonate formed? Bicarbonate is formed by those cells from carbon dioxide and water. So carbon dioxide reacts with water to form carbonic acid. That reaction is under the influence of carbonic anhydrase. And then it dissociates to hydrogen and bicarbonate. Then that bicarbonate will be secreted, and hydrogen will be utilized to maintain while the two major electrolytes in the body, the two major pH determinants, hydrogen and what? Bicarbonate. So when bicarbonate is going out, the hydrogen is doing what? Going in to keep the pH in order. However, if for any reason the blood becomes more alkaline, the hydrogen has to come out. But that will be done by the kidney. Alright? Okay. So for example, this is the protein. So that's the amino acid that make up. So exopeptidases will be cleaving from the outside. Endopeptidases will be cleaving from the inside. So those are the end amino and carboxyl group. So if they are attacked by exopeptidases, then the inside ones are attacked by endopeptidases. So now that we have known what the term means, so let's go back to carboxypeptidases. So they are, of course, it used to take place in the lumen, but the enzyme is from the pancreas, right? Now, that is the enterocytes. These are the enterocytes. And this is what we call the brush border. The brush border. And if this were to be the pancreas would have released what? Its enzymes. Right? Make sense? Now, we have peptides here, but not amino acids. So these enterocytes, on the surface of their membranes, they have enzymes. They now produce the enzymes that will attack these peptides, breaking it down to which will now be absorbed. That type of digestion is called membranous digestion. So digestion takes place at two phases: luminal digestion and membranous digestion. So the final breakdown of the peptides to amino acid by the aminopeptidases produced by the intestinal mucosal cells. That's those enterocytes, right? Will now break down will now liberate the amino acids. Okay, Helicobacter pylori.
I said I knew I was going to spell it wrong.
Helicobacter pylori. Segmentation. I keep drawing it as a straight line, but is it straight line? It's not straight line. By me looking at it, which muscle is contracting? Is it the circular or the concentric? Simple. So the contraction of the circular muscle, the contraction of the GIT, this is the GIT. Bulk is a very good stimulator of motility. That's why we are asked to take fiber. Put more fiber in the food so as to increase the bulk, so that if you have more fluid, can you see this is not touching the wall? So on the wall, there are, I think this one is finished. On the wall, there are what? Stretch receptors, which need to be excited to send information to the brain. So, there are stretch receptors here. So if you have something that does not really touch the wall, then the brain will not receive any information. All right? And ideally, it's supposed to give out motor fibers. So when this acetylcholine acts on this contraction happens. So if you have materials that are not bulky and do not distend the segment, you will not have motility will be low. But if you have something that fills it up significantly and even bulging it, it's better. It's better. Bulges it, then the receptors will be so excited, and then they send information fast, sensory input to the brain, and then the rate of firing will increase, the quantity of ACT will increase, and then the contraction will be powerful. So bulk is important. So that is why in diarrhea now, when water keeps seeping from the interstitium with the gas supported by the whole environment becomes bulky. Now you have the electrolyte, you have the gas, you have more water, and motility is going on at a very high rate. The push will be fast. And of course, it's watery because you've got so much water in it. This is for what? Osmotic diarrhea. But in endotoxic diarrhea, it is infectious driven. It's infection driven. So here you